Welcome to the UNC Core Center for Clinical Research Speaker Series. We appreciate you taking the time to join us for the presentation today. Our presenter is Dr. Liao. She is a rheumatologist and clinical investigator at Brigham and Women's Hospital and Associate Professor of Medicine and Biomedical Informatics at Harvard. She directs the P30-funded VERITY Bioinformatics Core and co-directs the Cardiovascular Clinic in Rheumatology. She also serves as co-director of the Applied Bioinformatics Core within the Massachusetts Veterans Epidemiology Research and Information Center. Her research focuses on integrating state-of-the-art bioinformatics approaches using clinical big data and rheumatoid arthritis outcomes research with emphasis on the impact of inflammation on cardiovascular risk. She's a leader in these areas and has spoken to various audiences, including several invited talks at the ACR. So we are very excited to have her present at today's CCCR session on applications of bioinformatics for clinical research in rheumatology. We will have a short break for questions after the first part of her talk. So Kat, take it away.
Great, thanks Amanda, and thanks for the kind invitation to come talk to your group and share some of our work. And let me share my screen. Everything look okay?
It does.
All right,
Yep.
Great. So today I thought I'd talk about applications of bioinformatics in rheumatology clinical research. And I'd like to start big, but then really dive down into really some of the details of how the rubber hits the road. So I'll start with talking about all the buzzwords out there: data science, bioinformatics, machine learning, natural language processing, and then dive into two studies that I've done with collaborators on how we might actually apply these two clinical research studies, and end with a slide or two about our CCCR and maybe talk a little bit about how we might link up some more. So I thought I'd start with the clinical question. As Amanda mentioned, my interest is in RA and cardiovascular disease. And over time, as I've been studying this, I realized we should know more than we do about heart failure, which is a leading cause of morbidity and mortality in RA. And in general cardiology, the field has advanced quite a bit, where treatment and management, it differs depending on what kind of heart failure you have, either heart failure with preserved ejection fraction or heart failure with reduced ejection fraction. And when we went to look at the literature, there were definitely some studies, but very limited studies on heart failure subtypes in RA, and even more limited data on whether we should be doing anything different for management. So the objective of this study was to study the association between inflammation and heart failure, as well as not just heart failure, but subtypes of heart failure in RA. And our hypothesis is that elevated inflammation will be associated with this HFpEF subtype more than the HFrEF. And the reason we think that is in the general population, it's been really hard to even phenotype HFpEF. But the going thinking is that it has a lot to do with inflammation more than HFrEF, which, as you all know, has a lot to do with post-MI and loss of myocardial tissue that leads to HFrEF. So what are some of the challenges here? There's obviously reasons why this hasn't been studied before. One of them is that studying heart failure requires an RA cohort with a sufficiently large cohort with both risk factor and outcome data. The second is actually defining the subtype. As I mentioned, HFpEF already does not have a great definition in the general population or in the larger cardiology studies. There's a lot of debate. And so for us to even start at this, we would need this ejection fraction data, which is not typically captured even in our large RA cohorts. Echo reports are available in sources such as the electronic health records, but they're in narrative form, so very difficult to extract. Otherwise, you'd have to review thousands of charts. Obviously, defining incident RA and heart failure outcomes is a challenge in any study. And so what I'm proposing here is that we look at a potential data source of electronic health record-based RA cohorts of 16,000 patients. I'm going to walk you through how we might do that in a few slides. So I'm going to just switch back to starting with talking about the broader topic of data science, which I think has the definition itself has evolved over time. The NIH describes it as an interdisciplinary field of inquiry in which quantitative and analytic approaches, processes, and systems are developed and used to extract knowledge and insights from increasingly large and/or complex sets of data. That was a mouthful. And so I went and tried to see if there were some nice figures out there to break this all down. And actually, University Berkeley, UC Berkeley, had this nice diagram of how they think about data science as different modules. You're capturing the data, often large amounts of data. So even trying to pull that in is a field in and of itself. Maintaining the data, building the right data architecture so you can pull the data and cross-reference as needed, cleaning, all that stuff is important for all of it. I know many of people on this call use a lot of big data and probably appreciate the importance of data cleaning and processing. Then there's actually the processing. So one is maintaining the data, then processing the data, and that has to do with how do you get data out of there. So data mining, summarizing the data, classification, and then actually after you pull out the data, analyzing, which is a lot of what we do with our modeling. And last but not least, communicating the data through visualization, and for us, it's research studies or research reports. Obviously, this field now crosses business, science, and medicine. And so you can see a lot of the jargon on the left you see in the figure is not words that we typically use in our work. And so for this talk, I'm going to focus on the areas where I think researchers really pick up, or where we tend to live in the field of data science, and that's in the process, the analysis, and in the communications. And I'd like to focus on clinical big data, which is electronic health records, and really just touch upon machine learning and natural language processing today. So I thought that I would start by giving you a layout, this tapestry of how we think about health records. And it's more than just electronic health records. But we often think of these data in two big buckets. There's structured data and there's unstructured data. Structured data are the type of data that are in a database and able to be extracted and is hard-coded. So for example, your ICD-9s, your electronic prescriptions. Then you have the unstructured data where you can have data in narrative notes. It can be in tweets, it could be in telephone notes. It's as you can imagine, unstructured, but there is information, important information you can get from there. So obviously I'm showing you here beyond electronic records, there are studies looking at tweets and other types of personal health records, but we're really gonna focus on electronic health records today. Next, I'd like to just introduce bioinformatics, which sits within the framework of data science. And so this is a definition from our colleague, Shomo Reishodri, who's here at the Brigham. And he defined bioinformatics as, and not just him, but many people, a way of combining algorithms and approaches employed in computer science and statistics to analyze, understand, and hypothesize about large repositories of collected biologic data and knowledge. And just again, give you an example of maybe 2 decades ago, or even a decade ago, the rate limiting step in biology was data collection, and now it's about interpretation. And I think the example here is where we went from, looking at single genes to all genes in simple organisms, and then exploding to high throughput technologies, beyond GWAS. Now we're looking at single cell technologies. And the example here I'm showing you is a figure I'm sure many of you've seen before, is a Tisney plot, just trying to visualize how lots of different type of data may cluster together so that you can take that and try to make some insights. So now let's talk about clinical data. So that this revolution has happened in genomic data and biologic data. I think I would say a similar thing has happened for clinical data. And that shift really, I believe, happened when we shifted from paper charts to electronic health records. In 2009, the US government passed the HITECH Act, incentivizing institutions to switch to electronic health records. So many of you probably recall in training back when we used to document everything on paper charts. And if you needed to find something about a patient that was admitted 5 years ago, you'd have to go down to the basement and flip through stacks and stacks of charts to figure out when they had that last MI. So you can imagine back in those days, it was very hard to study large populations. Definitely doable. They're very large, well done cohort studies, nurses health study, Framingham, just to give a few examples. But it wasn't as readily accessible, especially when you're trying to pull out many variables and outcomes. Fast forward to now with electronic health records, we have very high volume data designed clearly for billing, maybe for patient care, very suboptimal for research, but it does capture large amounts of data on large populations, and it gives us a lot of opportunity to think about how we might be able to use these data to understand our diseases better. And this is where I think bioinformatics, data science comes in, when you have this large amount of data to be able to take this and distill it down to what you need. Now I'm going to jump back to our clinical example, and that is looking at the association between inflammation with heart failure and its subtypes in RA. So as I mentioned, our hypothesis was that elevated inflammation was associated with heart failure with a stronger effect on HFpEF versus HFrEF. And one reason I am sure that we all care about as rheumatologists is that inflammation is modifiable. And so we should really think about, first, establish whether this is an issue, and two, it would help us in the future to understand how we might do a better job preventing heart failure, or specifically HFpEF. So this study was led by Selena Huang, who's a fellow and was a junior faculty here at Brigham. And I'm just going to start with the results, actually, and then circle back to under the hood. So what you're seeing here is our survival plot here, time on the x-axis, survival probability of heart failure. And overall, we stratified patients by those with elevated inflammation in this melon color, and in this teal color, and quote unquote normal inflammation in this orangey color here. And you can see that patients who had elevated inflammation at their baseline, this is around the time they were diagnosed with RA, had a higher risk of having heart failure. Then we further stratified by whether they had HFrEF or HFpEF. And so now you see HFrEF is in orange and green, HFpEF is in this blue and purple. And overall, you can see that most of the signals, so HFrEF is up here and HFpEF is down here. This baseline inflammation is more strongly associated with HFpEF, particularly those with elevated inflammation. And so in fact, when you look at it by tertiles, comparing the highest tertile of inflammation to the lowest here, you can see that predisposes patients to a higher risk of heart failure. And particularly, I'm going to go advance forward, definitely to HFrEF, but definitely HFpEF as well. And the blue and the red just have to do with time periods. We looked at both risk at 5 years and risk in 10 years, and you can see that the risk at 5 years is just as strong, if not a little stronger than 10 years. Okay, so now I walked you through what at surface looks like a classic clinical research study, but I wanted to take you to look beyond the hood and the steps prior to the analysis of how we got there. And I have to say, sometimes when I'm doing my projects, I think of this zero gravity cliffhangers. I don't know if any of you had this, it's this really old racetrack, electric cars, they supposedly would race around, but half the time the cars would fall off. It's a twisty road, they'd fall off and land flat in their backs. And that's how I felt throughout, actually I would say throughout my career, as we're trying these new technologies or trying to lay down these pipelines. So let's go back to how we started with the data and talk about the types of EHR data. There's structured data, as I mentioned before, ICD, CPT codes, things that you can really search for in a database, versus visit notes or unstructured data. And a lot of information here is, a lot of important information we need is here. What we do, just like any study, is you're extracting the data. And so you could query for the structured data. And for the unstructured data, we need to convert it into some form that we can analyze together with the structured data. And that's where natural language processing comes in. And so I'm going to just give you a very simplistic overview of natural language processing. One general way to think of it is a computational method for text processing based on the rules of linguistics. And so many of the NLP platforms, and there are many out there, will take a sentence and parse it into its parts. Some will, figure out what words they are or what type of words they are, and then try to interpret the sentence. And so this sentence says, I saw the girl with the ophthalmoscope. But as you can imagine, there's some ambiguity here. I saw the girl with the ophthalmoscope, meaning you saw the girl holding the ophthalmoscope, or I saw the girl with the ophthalmoscope, meaning, I saw her and I was using the ophthalmoscope. So there's a lot of ambiguity in sentences, and even more so, medical NLP is its own field because we write in abbreviations, we don't write in full sentences. So a lot of the principles of NLP have had to undergo some adaptation when taking it to the clinical notes. This is just an example of how NLP is more than a find command in Word. It looks at, it's able to say, so if I'm looking for erosions in RA, and this sentence says the patient has no erosions in the MCPs, it's able to say this is an instance where the patient does not have erosions, and I can say I only want to see instances where, or give me, tell me which patients had an erosion. Could look at inverted syntax. So if you're looking for colon biopsies, even that ascending and descending in between will allow it to pull this out as a colon biopsy. There are more complex uses of NLP. I have to say we use NLP very simplistically, but they can look at relationships. So this is a simple one. Tamoxifen is used in the treatment of breast cancer, but there are studies that use NLP to look at relationships between drugs and adverse outcomes. And last but not least, morphologic variation. So for example, smoking is a very important risk factor for RA, but it's written in many different ways. And there are standard sets that try to account for all these morphologic variations, and there are ways to tailor what other variants you might want to pull out of there, so that at the end, you want to create a smoking variable, yes or no. All those different ways of saying smoking will pour into a smoking, what we call a feature, yes or no. And so now we have the structured data, we've processed the notes, we've pulled out features and concepts that we think or we would like to study that are important, such as smoking erosions. And now they're converted into numbers as well, so that we could analyze them together. And so there are many approaches for phenotyping, phenotype algorithms using EHR data. I'd say many of us probably started with chart reviews, but when you're looking at these populations, these EHR populations of millions of patients, chart review is just not feasible. They're then rules-based, and this is still very useful, particularly in claims data, but it does rely on human expertise to identify the important features. And these rule-based algorithms are usually a combination of and, nots, and or. So 2 ICD codes and a medication, or 2 ICD codes X times apart. The last method, and I think the one that many of us have been testing to see if it'd be useful, is a data-driven method to select the features, meaning what goes into the algorithm, and or mostly develop the algorithm. And so, features it can be automated, or you can input them in, but the algorithm itself, when you decide which of the important features or variables, that's often the point where we're applying a lot of machine learning. And I want to say I'm focusing on EHR data, but I know that this group is no stranger to using machine learning for phenotyping, and it doesn't have to be EHR data. It's really just very useful for very large data sets, especially as we think about phenotypes beyond just RA. There are subphenotypes beyond OA, and there are subphenotypes when you think about OA or progression of OA. So I wanted to just briefly introduce you. So over time, for phenotyping, this is a prevalence, yes, no, in the EHR. We've developed a pipeline, which includes machine learning. And so we start with the EMR, and the first step is a filter where you can say, okay, I want everybody with at least one RA ICD code, because anybody with no RA ICD codes, it'll be nearly impossible to ever know if they have RA. So you just kind of have to assume that they don't have RA. The caveat there is that a code like RA is highly sensitive and not very specific, which is great for a filter. So you're trying to pull everybody who can possibly have it. Next step you see here is we create this data mart where we have the structured data, such as ICD, electronic prescriptions, and the narrative data. We extract the data using ICDs, NLP, apply NLP, pull out these data, and then this is where a lot of our machine learning methods take place. I'm not going to go into too much detail, but we now do a two-step process where you do unsupervised feature learning, meaning, sometimes you end up with 300 features, potential features. You still need to whittle it down to maybe 70 to 100 before you try to train the algorithm on your chart review labels, because otherwise you'll be completely overfitting your model. So we have a two-step process here where we do unsupervised feature learning, meaning we try to whittle down from 300 to, let's say, 50 features. And then we do a supervised algorithm where for many of our algorithms where we're creating a cohort, we have a gold standard set where somebody has chart reviewed 200 charts and identify whether or not, for example, they had RA or not. One of the major differences of these machine learning algorithms, or this new approach for algorithms, is, if you recall, the first example I gave you, the second example was rule-based. 2 ICD-9s and a prescription. It's yes or no. People are in or out. Machine learning algorithms give you some more flexibility. So our algorithm at the end is a logistic regression model that spits out a probability of RA or any other phenotype for any patient. So you take your 2,000 patients, or for in our instance it was 5 million patients, and everybody gets a probability of RA from 0 to 1. And so from there, you can threshold, you can say, Well, I know based on my training and validation set that above a probability of 0.5, let's say, everybody has a positive predictive value of 90%. But let's say I'm doing a pharmacovigilance study. I want more patients. I'd rather err on the side of some misclassification, but I want to capture the larger population. Well, maybe then you take a lower probability and you threshold at, let's say, 0.45, and now you have a PPV of 85%. And so that gives you more flexibility to design your study, kind of get a sense of the power and how many patients you might have in your final analysis. And this is just an example of what the algorithm looks like for actually RA. PA just stands for phenotype A, and it's the machine learning process that helped us to weight and decide which features end up here. But at the end, it's a very kind of algorithm that you can understand what went into it. So I'm not saying that all machine learning algorithms come out this way. Some of them are black boxes. And so as someone who's a clinical researcher working closely with the bioinformaticians, we really strive to translate these machine learning models into models that someone like me would, I'd say, okay, I feel okay applying this. And so we used PheCAP, or this pipeline, to develop an algorithm from RA and heart failure. So what does that really look like? We took EHR, we first classified RA, and then within RA, we classified heart failure. And this is just an example of the performance of heart failure here, and we were able to achieve a positive predictive value of 90% with relatively good sensitivity and specificity. Again, using this published pipeline that we've actually tested across maybe about 20 phenotypes now across 3 or 4 institutions. So the next step is just to give you a flavor of what else is out there, what other tools are available to us. And so the other thing that we challenged that I pointed out in the beginning was, how do we get ejection fraction out of there? Even though we've now limited it to our 16,000 RA patients, there's probably still about 10,000 patients with echo records where it's not easy to pull out the EF. It's not hard coded. You can't do a search or query saying, give me the EF, and you get it. And so this is just an example of this study was led by Tianwen or Austin Kai in our group. He's an expert in NLP, and what he did is he created a tool to extract ejection fraction from the notes. And so just very briefly, he takes the EMR notes and he created a pipeline using some of the existing toolkits. You see the natural language toolkits here to extract data such as vital signs and EF. And what is this really doing? It's normalizing and tokenizing. So it's taking a large amount of text data and breaking up into its parts. This is a rather manual, I would say, a rule-based process, meaning EF is often written in so this kind of tool is very useful for numeric features that are written in very similar ways. So pulse, fever, ejection fraction, all the thing that's common across them is that they're often written in very similar ways, and there's a range that they can be. So what he's doing is he's normalizing and tokenizing these narrative data with this natural language toolkit. And so I'm going to give you the example of EF here. Here he's looking for TTE, EF 21, and he's kind of specifying, well, what are some of the possibilities that you're looking for. So now, and we validated that with EF, it did quite well. And now you have your RA, heart failure, and within heart failure, actually with everyone, we just ran extend, which is a tool, and pulled out everyone's EF. And so now we've got a cohort of 16,000 patients. We did, in terms of incident RA, use the first RA ICD or NLP concept. We extracted actual lab values from the EHR for ESR and CRP. We pulled out covariates. This is using ICD codes. And the outcome, as I mentioned, heart failure was classified using the algorithm with 90% PPV. We extracted EF data. And then we defined incident heart failure, again, using a rule-based process, where it turns out the first heart failure ICD or Lasix or furosemide NLP, whichever was later, turned out to be a pretty good proxy for incident heart failure. Next, we designed the study as you would any epi study. You want to think about, what's your exposure window and covariate window. So we designed the study to say, Okay, you have incident RA. We want to look at your covariate window for risk factors. We have a window for exposures, and then this follow-up window here until you get heart failure. And so we found overall 9,000 RA subjects with 1 year of data prior to their first RA diagnosis, because we wanted to know that they didn't have this incident RA, incident heart failure. Mean age was 56, 77% female, 55% seropositive. Of this group, 749 developed heart failure, of which 561 developed HFpEF and 127 HFrEF. And you'll notice that, you know, we did, there's also a HFmEF category, but I think even now people aren't sure exactly what to do with that category. So we focused on the two extremes. And this is the figure I showed earlier. Again, this higher risk for HFpEF, particularly with inflammation, but heart failure in general. And you'll see that we did also adjust for multiple covariates. And so in conclusion, we found that HFpEF was 4 times more common in RA versus HFrEF, which I thought was a surprising number. It was more than I thought, but it is in line with the few studies we have out there, which studied HFpEF and HFrEF using ICD codes. Elevated inflammation is clearly a risk factor for heart failure, independent from the traditional risk factors, and the signal is really driven by HFpEF. And I hope this gave you a nice example of how we've integrated data science, machine learning, NLP in the framework of an epidemiologic study, in the framework of how we continue to do studies to study a clinical research question. And so I'm going to pause here for any questions before I jump to the next example that I'd like to show on another bioinformatics platform.
Who is your team for this, Kat? How many people are you utilizing the skills of in order to do this work?
That is a very good question. So I have a relatively small team at the Brigham. So it's me, junior faculty, as I mentioned, Austin Kai, who's an NLP expert, data scientist, a database programmer, and a research assistant. So that's my core team. This is the core, my lab, which also happens now to be the bioinformatics core. But I've been very fortunate to have a longtime collaborator who is a biostatistician and bioinformatician. And she has a lot of postdocs who develop these methods. So maybe I should say the core study, this heart failure study I showed you, are pretty much those 5 people. And plus, fellows, cardiology fellows, other people. But in terms of the earlier stuff I showed you with the methods development, that is in collaboration with another group at School of Public Health and Harvard Medical School, Biostats and Bioinformatics.
Can I ask something? I know that you have R package, PheCAP.
Yeah.
So do you have any, opinions from users outside of your institute, how easy it was to use, the package itself? If you have package, if you have data, how easy it is to implement?
Yeah, well, thanks for that question. So yes, what we strive to do is when we create a pipeline and publish it, what we often do before we publish is we try it out in another institution. So I'm fortunate. So I should answer Amanda more realistic. I have like many hats. So I'm based here at the Brigham, but I also co-direct a bioinformatics core at the VA. And the VA is very interested in, broadly trying to get these things to work. So what we've been doing is taking our methods that we developed at Brigham, and that's where they go to VA to get broken and beat up. And so the R package for PheCAP, we took it to the VA and tested it there. And that's when it became more of a pipeline, and we wrote a package and methods paper. So we do try to design it so that it's that it will run anywhere. And for example, PheCAP also comes with these toy data sets to show you how the data sets should be formatted. And so, you could reformat your data set to plug it in.
Is it possible to extract information like in safe use, not the outcome like heart failure, but something like more, risk factors like over-the-counter medication or something like that. Because, it is not always available in this, database, but from other sources.
Right, yeah. So so I would say, just like any tool, there's only a certain range of uses it's for. And so something like PheCAP, right, it's really, it was designed and tested on phenotypes that have ICD codes and that are kind of classic conditions. There are lots of things out there, like you're mentioning, NSAID use, that are just really difficult. Smoking is another one that's really difficult to capture. And I can't say, we don't have a pipeline for that. And I'm not actually aware of any other pipeline. I'm sure you're more aware of the methods, different methods used to try to extract that data.
I had a quick question. As a rheumatologist, how did you gain this skill set? And what would you recommend to fellows, who are interested in academic research and similar type of research as a roadmap, getting a master in public health? And I guess, what would you recommend to the fellows who are interested in similar work?
Yeah, that's a good question that I get a lot. And the field has changed so much. So I can tell you how I got here, but it isn't what I would necessarily recommend for the next generation here. So I was really fortunate as I was coming through fellowship. I had started doing work, with just our clinical epi studies. And at the time, there was this big study starting up. So talk about the HITECH Act, right? 2009. One of Zach Ohani, who's now the chair of the Department of Biomedical Informatics, he said, this is a big opportunity. We should figure out how to use EHRs for research. So he wrote this big grant, and he said, I need people as guinea pigs. And so I was like, okay, I'll try it. And so I got embedded in this team of, NLP, bioinformaticians, biostatisticians, database programmers. And together, there was no roadmap. We were like, how do we even do this? And we'd sit in a room for 3 hours once a week and just try to figure it out. So let's fast forward to now. So that's how I got my skills, because there were no courses. Fast forward to now, I can tell you Selena she did her fellowship, and then there was a master's in data science at our school of public health. And so she did that, and then obviously did some hands-on projects with our team. And that's how and I would say that would be probably the best way now to get the skills. I do want to say that because it's still relatively new, you really need to look at the courses that are offered, because bioinformatics, as I mentioned earlier, has many different meanings to different people. And some bioinformatics courses are very genetics oriented. There are fewer for clinical data or just general methods. So that would be the caveat. All right, should I jump to the second? Maybe I'll jump to the second portion now. Okay, so I thought I would share another tool called the PheWAS, which many of you may be familiar with. It's the Phenome Wide Association Study, and it's one of those things that wasn't necessarily, the idea, like the pieces of it aren't super exciting, but this concept wasn't really available until we had EMRs and biorepositories linked together. So many of you are probably aware of all these biorepositories, right? You got UK Biobank, you've got MB Million Veteran Program. And so I guess one of the questions, what's the buzz all about? One thing is that when you link the EMR with a biorepository, now you have a lot of phenotypic data, and you're able to have the genetic data standing there. I don't know, many of you probably recall way back when, or not that long ago, when you wanted to do a genetic study, you had to carefully select your cohort, genotype them, and then you had the genotype and phenotype, or you're recruiting patients for 10 years to get enough RA patients and then running that GWAS. Well, now you've got 500,000 people sitting there with their EHR data and their genetics already there. And the challenge is now on the phenotype side to try to find your RA patients. I didn't go into detail on this, but finding RA patients you would think is easier than it is. But just to give you an example, the positive predictive value for one RA code is about 20%, no matter where you go, if you do an unselected sample. Or I should say that was the case in Brigham. It was the case when we went to VA. It was the case when we looked at Northwestern as well. So let's talk a little bit about a PheWAS. So I think you all are aware of, probably all aware of what GWAS is. So thought I'd start there. On the x-axis, you have the chromosomal position. On the y-axis, you have the negative log P, and we all know that shared epitope is our top hit here. So you're looking at millions of SNPs and one disease, yes or no. So let's take a PheWAS, and what we're doing is we're flipping it on its head in a way. So as I mentioned, now you have the phenotypic data and the genotypic data together. So this group, Josh Denny and his group, who's at Vanderbilt, and they really, I would say, he doesn't want to take credit for the idea of PheWAS, but they've really enabled us a way to standardize PheWAS. He said, okay, so HLA-DRB1, so important for RA, but what else is it important for? And so when you have these data sets, you can now take the SNP and then do the associations, do multiple logistic regressions to see if it's associated with any of these other phenotypes here. So now you have phenotype on the x-axis here, negative log P on the y, and you see that there are these other diseases. RA is a positive control here that where HLA-DRB1 is important. So the phenotypes themselves are classified. So Josh Denny and his group, they went through all the ICD codes and grouped them into what they considered clinically relevant groups for genetic studies. So you can see that type 1 diabetes is still separated by ketoacidosis or a general category. But as you all know, there are these much smaller categories that still require grouping in. So I would say MI is probably a good example, myocardial infarction, where you have like anterior, inferior, you just want to put that all as MI. Otherwise, you're just increasing the number of comparisons you do and losing power to detect an association. And so I thought I'd walk you through a PheWAS that we did at the VA in collaboration with the Million Veterans Program. And what I was interested in as a rheumatologist is IL-6R. This is interleukin-6, a pro-inflammatory cytokine, important for lots of things, especially treatment for RA and now GCA. So at the time when we started the study, the IL-6R tocilizumab was actually only indicated for RA and not yet for GCA, but now it is. Cardiologists are also really interested in this variant, and they are even more so now. There are randomized controlled trials now looking at IL-6 blockers. But back then, there was great interest because it seemed to be associated with lower risk of coronary artery disease or coronary heart disease. The interesting thing about this variant is that we know the function. So individuals with the variant, so you're not on an IL-6 blocker, you have biochemical properties similar to those on IL-6R. So people with this variant have lower CRP and fibrinogen concentration, which is the same thing you would see with somebody on tocilizumab. And then, as I mentioned, lowering CRP, there was a lot of interest in cardiology, and there was a Mendelian randomization analysis that showed maybe there's a potential causal association between having this IL-6R variant and reducing the risk of coronary heart disease. So the other thing about this drug is that it had a gene-drug pair, what I call a gene-drug pair. There was a variant that kind of acts like the drug, and we have a drug out there already that we know what it does. And so this is something that will give us the ability to potentially use genetics to query potential drug effects, and with implications for drugs in development where we know the function and there's a variant that kind of acts like the drug. And so IL-6 here, binds to the IL-6 receptor, and this signaling pathway leads to inflammation. Sarilumab and tocilizumab block this receptor. And so this variant, what it actually does is it results in downregulation of IL-6R. So you have less of the receptor and thus less ability to signal via IL-6. And overall, how I think about this, so you might think about this, is that it's like you're on a little bit, people with this variant, it's like they're on a little bit of tocilizumab or sarilumab their entire life. And so the implication of this is, if you're on a little bit of tocilizumab, what would it look like? How will your phenotype differ from others? How will the people who seem to be on a little bit of tocilizumab their whole life because of this variant differ from those who don't? And so what we wanted to do in this study was to do a proof of concept. We wanted to look at this IL-6 variant and see if we can capture known on and off targets of the IL-6R antagonists. And in this case, because tocilizumab had already been out there, the sarilumab RCTs were out, we can compare PheWAS findings with published results as a way to think about how we can use this tool to query potential drug effects. And so just to summarize again, we have our drug tocilizumab, which binds to IL-6R and inhibits IL-6 signaling, and the in vivo effect in humans is the drop in CRP. For people carrying this genetic variant, they have a reduced expression of this membrane-bound IL-6R, which in the end inhibits IL-6 signaling and also drops their IL-6. So kind of a match here. It's kind of my little checklist. Now you have this drug, a genetic variant match, and now you have the EMR. We have, at the time, 300,000 veterans with genotypic data, and we can look at the association of people who have the variant versus those who don't, and the EHR phenotypes. We performed this study in the Million Veterans Program, as I mentioned, 340,000 at the time, and extracted these information on all subjects, IL-6, ICD codes. We also wanted to look at routine laboratory measurements, which is one of the, I'd say one of the advantages of having an EHR oftentimes is that you can look more than just whether a lab was ordered, you can pull out the lab results. And a particular interest to cardiology is whether or not there's any difference in cardiac troponin or CRP. So the ICD codes were mapped to Phe codes. And again, this is a standardized mapping that was established by the Vanderbilt group, and they continue to update it to ICD-10. And I give you an example below of how you might just pull acute myocardial infarction together into one Phe code. So you don't have to specify which wall it's on, because for a study like this, that's too much detail. Then we do a logistic regression, and what you're looking at is the association between the variant and the Phe code, yes, no. So there are about 1,800 Phe codes out there. For the laboratory values, we're taking the association between the SNP and the laboratory values in linear regression here. And for significance threshold, we use both the false discovery rate and Bonferroni. In this case, Bonferroni is probably a little too strict because there's a lot of correlation between the Phe codes and the labs. So we ended up reporting both. So among the significant associations, we viewed charts to estimate the accuracy of the Phe codes. As I mentioned earlier, the accuracy of ICD codes vary widely. So you want to know exactly what it is. Once you get a hit, you want to know, well, was it actually the phenotype that we thought it is? And so we did do chart review in addition. And we additionally validated any of our findings in biobanks with freely available data, and at the time, UK Biobank and BioView. And fortunately, a proxy SNP that was highly correlated with our SNP of interest was available in both data sets. And so this is our finding here. In the end, we had 330,000 subjects. And again, on the x-axis here, you have the phenotypes, and this is grouped by the ICD codes. On the y-axis, negative log P, and we're looking at that one SNP for IL-6, one genetic variant for IL-6R. Triangles pointing down means reduced risk. Triangles pointing up shows increased risk. So maybe I'll start actually on the right here with hemoglobin. I was happy to see this because I think maybe 20 years ago, there were oncologists were interested in IL-6 and cancer. And they literally infused humans with IL-6 and saw that their hemoglobin dropped. And then when they stopped, their hemoglobin went up. And that had to do with some increase in fluid in the intravascular system. So in this case, when you block IL-6, again, you expect an increase in hemoglobin from that functional study, and we're seeing that here. We also saw the results that you would have expected to see from the Mendelian randomization analysis of IL-6R, where individuals with this genetic variant had a reduced risk of coronary athero. And as I mentioned earlier, when we embarked on this study, there was the trial for IL-6R for tocilizumab and temporal arteritis was underway. And what we saw was a signal for reduced risk of aneurysms with IL-6R. So overall, we did see some expected associations that you saw from prior studies, potentially new associations. And I think one of the things that actually I was surprised in is you're looking at one SNP and potentially kind of noisy phenotypes from the EHR. We all know how good the ICD codes are, and the fact that you can capture strong signals. And I would say the caveat is you probably can't capture any signal that's weaker. This blue line here is the false discovery rate, and the red line here is the red line here is the red line here is the Bonferroni for us, and the blue line here is the false discovery rate. We also took a look at the markers. So again, this is biased, right? This is not technically what I would call a PheWAS, because it's for a PheWAS, you want to have everybody to have the same measurement or the same opportunity to have the measurement. In this case, we're only looking at measurements that were done as part of routine care. But what we did see is lower CRP, people with the variant had lower CRP, lower troponin, and lower BNP. And so some of the strengths I'd say is, this proof of concept, we're really able to capture signals, leveraging knowledge of the gene-drug pair. And that actually, I didn't show you the replication, but we were able to replicate the cardiovascular, the aneurysm, and atopic dermatitis in a UK biobank and BioView. And just a quick note here that MVP is working hard to get their data out there. Some of the limitations, the obvious limitation, RA, is why I embarked on looking at this treatment, and it was not identified by the PheWAS. So one thing to keep in mind is that the prevalence was low in the VA with 3.2% for the code. The accuracy of the code, and so if you take two codes, had a PPV of 35%. So we just did not have the power to detect an association. We also did not catch GCA, which was what the New England Journal trial showed. Again, the prevalence of a GCA code was 0.45% with a PPV of 65%. And aortic aneurysms as a complication from GCA was encoded within the aneurysm code. Overall, the phenotypes range in accuracy from 35 to 100%. And some of the work that my larger collaborative group is doing is to understand or to develop methods to phenotype in a high throughput way, meaning it's just not possible to do chart reviews for thousands of patients. But how do we integrate NLP and ICD to just boost the PPV of all these codes? And last but not least, there's not a clear threshold for what a significant association is. And there's possibility that you might miss some, and obviously that you might pull in associations that are not real. So actually, I thought I would pause here. Actually, you know what? I'll just give you the last two slides about VERITY, and then I'll just leave the rest of the time for questions. So I wanted to briefly introduce our core at the Brigham. It's called VERITY, and it's NIH P30, much like yours, and show you some of the faces of VERITY. So Dan Solomon, who's also chief of the section of clinical science here, is our overall PI. We have three cores, admin, methodology, and bioinformatics. Methodology is led by Yelena Lucina and Jeffrey Katz. Bioinformatics, myself and my longtime collaborator, Tianchi Kai, who I mentioned earlier. And we also have a clinical research course coming up in May, directed by Jeff Sparks, and also Jackie Stratton is doing all the coordination for that course, and actually for many of our cores. So I did want to put in that plug where we will be covering many of the concepts of clinical research across the span. You could take a look at the agenda, as well as there'll be a few talks on bioinformatics for those who are just trying to get their feet wet in clinical research. And so I'd like to thank, I had discussions, how big is my team? Well, this is my team here, and some people have come and gone. This is the team over at DBMI, I'd say ending at Amanda King here. We meet together twice a week sometimes, or actually we do meet together twice a week for various projects. And this is the team over at VA that we collaborate. So lots of meetings, lots of work, but a lot of fun having all different kinds of input. So I've been very, very fortunate to have particularly Tianchi as a longtime collaborator, and then folks at the VA to really, develop things, go for the whole spectrum from thinking of the clinical question, developing the methods, employing it in a clinical research study, and then potentially scaling up to see how we can share this with others and other institutions. So I will stop there. I want to thank everyone again for allowing me to share some of our work.
That was fantastic. Thank you, Kat, for sharing that. I just want to let you know that even though the crowd here is a little small, our fellows usually watch this afterward because they're all in clinic. So I'll be sharing it with them. And we do have somebody that may be interested in the VERITY course. So
yeah, they should totally sign up
about that.
Yeah.
Yeah.
So I'm also happy to hear some
usually
ideas.
This quiet.
Yeah, well, I'm also happy to think of some ideas how we might link up more, with the cohorts. So
I just want to say that was a fantastic talk, Kat, and I'm like exhausted. You have so much going on. It's amazing. I'm like Amanda, like I was thinking, like, yeah, who all is doing this?
Yeah, I guess I'll just throw some stuff out. So we obviously are interested in osteoarthritis. It's horribly coded, right? So most people that see it don't bother to code it. And when it is coded, it may or may not be the actual disease. So I think there's been, to this point, a little bit less enthusiasm about using EHR data for OA. And I think it's really going to have to be much more natural language processing and like pulling osteophytes out of radiology reports and that kind of stuff, but obviously much more prevalent. So I just wonder what your thoughts are on sort of the logistics of making the leap from something like RA that's at least usually there when it's coded versus OA that maybe is not, there aren't any medicines for.
Yeah, I would say it's probably similar. We work with many different people, and it's like that we've come across phenotypes that are poorly coded that have more description in the charts. And so you're completely right. It's still EHR data, but you're going to lean heavily on the unstructured data and the notes. And so there are definitely methods there to try to, use beyond textual data to actually take the concepts out of text data, right? Like think of clustering. And then instead of taking ICD codes and clustering, now you're taking text data, osteophytes, joint space narrowing or cartilage loss, putting them into concepts and clustering by words. So, one thing that comes to mind, and I don't know if that will be the right way, but topic modeling has become really useful for us. An example of topic modeling is if you were to take the New York Times and topic model and tell them to group words into clusters, you'll see that it'll roughly group into like sports type words and politics type words. And so it just kind of gives you a sense of how things go together. And then you can put those clusters into the model. So that's one thing that's been helpful for phenotypes without ICD. I'd say the second big thing for OA might be, and this is an area I have no expertise in whatsoever, is imaging. There's imaging data, taking that and performing machine learning on that to look at the different phenotypes.
Anybody else? All right, I think they're all processing the large amount of information that was just thrown at them. So, we really appreciate this, Kat, and I'm really glad to have you talk to our group, and then we'll share this again with some of our trainees, and I'm sure I at least will be back with you on some more brain picking.
No, it was really nice to meet you all and see you all virtually. But thanks again for the invitation.
Thank you again.
Good.